What's up, payments? Today we're going to do 2024 number 1, cell cycle crossing over and experimental design. This one has a national average of 3.09. Now this is question 1, which means it is one of our long FRQs, and they changed the format last year in which they're going to have prompt, question, and then some more prompt, and then some more questions. So they did break it up. So do bear with me as we kind of alternate between a long section of reading and then questions. So the first part, crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. So part A sub I, describe the function of the S phase of interphase. So as a reminder, interphase takes place before mitosis, and mitosis are nuclear division. And when we go through mitosis, we need to have identical cells. So in interphase, we need to replicate our DNA in some way so that we can have 2 sets of S chromosomes. So the function of my S phase is for us to go through that DNA synthesis. We can synthesize a copy of our DNA. And so the function of the S phase is replicate, duplicate, synthesize DNA or chromosomes. You could also say that we're doubling the amount of DNA. So what the students say? Students said the S phase of interphase is where the DNA is replicated in preparation for cell division. The helicase enzyme will come and bind to the origin site, unzipping the 2 strands of DNA. Then RNA polymerase will build primers where the DNA polymerase enzyme can synthesize both the lagging and leading strands. By duplicating its genome during the S phase of interphase, the cell then has 2 copies of its DNA, which then can be separated during cell division. So they did mention that we were going to have 2 copies, which was what was in the prompt, which was in the scoring guideline. But they have already gotten that point for saying the DNA is replicated, so they wouldn't get an additional point. But that was actually a great response, giving very thorough answers for our describe. So here we have some more prompts. Some regions of a chromosome called hotspots display a higher frequency of crossing over than other regions do. Crossing over is suppressed in chromosomal regions near the centromeres. The centromere region of a duplicated chromosome includes a collection of proteins that form a structure called the kinetochore. Scientists hypothesize that one or more of these kinetochore proteins are responsible for suppressing crossing over around the centromere. To investigate their hypothesis, scientists modified chromosome 8 in yeast such that in each cell, one chromosome has a pair of homologous chromosome 8s contained the gene encoding red fluorescent protein, while the other of the pair contained the green fluorescent protein. Cells emitting the green fluorescent protein emit red light, while the cells expressing the green fluorescent protein emit green light. And we have models of the chromosome 8 both before and after crossing over in our given figure. So I did color code them just to make it a little bit easier to follow. And so here we have our chromosome that has our red fluorescence, and then this one has our green fluorescence. And so after crossing over, we can see that this top one did not have crossing over, so it still has the red. This one had crossing over and it lost its red fluorescent protein, but this one right here was the one it crossed over with, and so now it has green and red. And then this one on here is just our green, and so the fact that it has, of course, did not have crossing over, so it has a green fluorescence. And so as you notice, these ones in the middle were where their crossing over took place. So the question says, explain why some haploid cells formed after meiosis in the experiment will only have 1 fluorescent marker. And so as we already mentioned, the interior ones were the ones where we saw crossing over taking place. And so due to crossing over, maybe they only have 1. You could also talk about that during meiosis, we're going to only have 1 of these chromatids in that daughter cell. So the options you could have here, some cells will receive a chromosome that did not go through crossing over. So those were these ones we saw on the outside. They did not undergo crossing over, which allowed them to only have 1 fluorescent protein. 2 crossing over events occurred. So if we had 2 of them occurring in that hotspot, that could lead to there being only 1 set or 1 of the genes. Each daughter cell will only get 1 of the 4 sister chromatids, and 2 of the ones that are shown in figure 1 are already going to only have the 1 fluorescent protein. And 2 chromosomes that were not involved in crossing over will only have the green or red fluorescent protein. After meiosis 2, a gamete with 1 of these will have either 1 or the other gene. So the student said some haploid cells formed after meiosis will only have 1 fluorescent marker and not both, because not all chromatids experience crossing over. In the 2 homologous chromosome 8s, only the 2 close together chromatids experience crossing over, where the other 2 didn't. During meiosis 2, when the sister chromatids separate to create haploid gamete cells, some will have chromosomes with both markers, and some will have neither, and some will just have 1 fluorescent protein marker. And so as you notice, they did have 2 parts that were scoreable, so they got their 1 point here because they've stated one of the things. So we have more prompt. Scientists then investigated whether attaching individual kinetochore proteins to a specific DNA sequence present in a known crossing over hotspot on chromosome 8 affected the frequency of crossing over at this location. In their first experiment, they examined 3 groups of yeast cells containing the modified chromosome 8. Group 1 contained no kinetochore marker proteins. no kinetochore proteins attached to the hotspot. Group 2 contains kinetochore protein CTF attached to the hotspot, and group 3 had the kinetochore IML attached to its hotspot. For each group, the scientists determined the frequency of crossing over between the genes. To determine the frequency, they added the number of cells emitting both red and green light to the number of cells that emitted no light and divided by the total number of cells. And so here you can kind of see that graph. You can see that the none has a higher amount. It does overlap its error bars with IML, and then the CTF does not have any crossing over, and it has a lower amount of crossing over. So B1 says, or BI says, identify the control group for the scientist's first experiment shown in figure 2. So what we're seeing is that which one of them does not have the treatment group? Well, since this first group right here has none of those kinetochore proteins, then that would be our control group. So group 1, the cell that contained none, or the cells with no protein attached to their chromosomes, okay? Because they don't have that experimental group, that treatment group. So students said group 1 with no kinetochore proteins attached to the hotspot is the control group for the scientist's first experiment. So then B sub II, in the follow-up experiment, scientists created a modified version of CTF in which the DNA binding portion had been removed. They compared the frequency of crossing over in yeast cells in the presence and absence of unmodified CTF with that in yeast cells in the presence and absence of the modified CTF protein. In the follow-up experiment, justify why the scientists used the modified CTF protein that is unable to bind to the DNA as a control. So using the modified one is going to allow us to compare to see whether it's inhibiting our crossing over, whether it's going to change how much crossing over is taking place. So to determine whether just the presence of the protein is enough to inhibit the crossing over or modify the recombination frequency. So we need to see, did that specific material in there have an effect? And so they said the scientists used modified CTF protein that's unable to bind to DNA as a control because it can show them that the CTF protein binding of the DNA that caused the decrease in frequency of crossing over and not another variable. So they were actually seeing that it did connect. So then three I's, identify the independent variable in the follow-up experiment. So looking at the type of CTF because they were using the presence and the absence of unmodified CTF. So looking at which type of CTF was used and which one was present is the variable that they were using there. And so that's what the researcher was changing the experiment. As a reminder, independent variable is what is being changed, and the dependent variable is what changed because of the researcher changing something. So the independent variable in the follow-up experiment is the presence or absence of either modified or unmodified CTF protein. So then based on the figure 2, describe the effect on the frequency of the crossing over when the CTF is attached to the chromosome 8 hotspot compared to the effect with the IML is attached to the hotspot. So as we said before, let's compare the CTF and the IML. And so you can see that it is lower. There's less frequency, there's less crossing over taking place in the CTF. And you can notice that the error bars do not overlap. Because the error bars do not overlap, the data is statistically significant. And so we can say that there is a decrease in the frequency of crossing over when the CTF is attached compared to the IML. So CTF results in a decrease or a lower frequency of crossing over. IML attachment results in an increase or a higher frequency of crossing over. And so the student said there is a decrease in the frequency of crossing over when CTF is attached to the chromosome 8 hotspot compared to the frequency relatively not changing because the error bars overlap with no protein group. When the IML is attached to the hotspot, the CTF's frequency is less than when there was no protein attached, whereas IML's crossover frequency is almost the same as when there is no protein attached. So part D, predict the effect on the number of copies of chromosome E likely to be present in the resulting daughter cells when CTF is attached to that hotspot. Okay, so if we're seeing that there's a decrease in our number of crossing over, okay, then there should be 0 or 2 copies, or there would be 1 less or 1 extra copy because of the fact that if we're not having that crossing over taking place, the logic is that we're not going to be able to have them separate during meiosis. And so the student actually says when the CTF is attached to the hotspot, the number of copies of chromosome A is likely to be different. They just said different and not the usual 1 chromosome 8 per haploid daughter cell. So they understood something, but they didn't clearly state that it would be 0 or 2, and they didn't talk about 1 less than 1 extra copy due to that Non-disjunction. And so the student actually did not earn a point in this because it did not predict a specific difference in the number of copies of chromosome 8. So they did not actually get this point. So then the second part, this DII, provide reasoning to justify their prediction. They did justify. They said that the cells were undergoing the crossing over at a lower frequency because it's more likely that the non-disjunction would occur and the cells would not separate properly. And so they said this is because crossing over is required in meiosis 1 for homologous chromosomes to be properly aligned. Since CTF protein decreases the frequency of crossing over, there is then a higher chance that the chromosomes don't align properly, leading to a potential non-disjunction events where some daughter cells can get more or less chromosomes than normal. So they clearly understood what was happening, but they didn't explicitly state it for the D sub I, so they did not get that point. So sub III explain how the presence of hotspots could increase the likelihood that a population would survive in the presence of selective pressures. So the hotspots allow there to be an increase of crossing over. And so we need there to be crossing over because crossing over increases our genetic diversity. Okay, so hotspots would increase genetic diversity. Therefore, it would be more likely that some individuals would survive and reproduce. So we do have to connect that back to the selective pressures and back to evolution. And when we talk about natural selection, we're always talking about the things that are going to be more likely to survive and more likely to reproduce. So you always have to bring it back to that whenever talking about selective pressures. And so the student said the presence of hotspots would increase the likelihood the population would survive in the presence of selective pressures because crossing over leads to genetic variation. Hotspots where crossing over happens more frequently help mix up the alleles of the chromosomes, resulting in each haploid gamete carrying a completely new and different genome. With this increased genetic variation, populations are better able to adapt to selective pressures because one individual might be fitter than another. So hope this will help. Remember, AP Bio Payment just satisfy all.